understand the molecules that are playing the roles that we're talking about.
So thank you very much, Gail. My thanks again to Ned and to Janice and Stephen and Gail and the other speakers who we'll hear from after the break. So my job is to talk to you about the structural biology of checkpoint antibody therapies from the perspective of someone running the global protein data bank, US data center here at Rutgers. So very briefly, I will introduce the PDB, I think well known to some of you and not others. Then I'll actually show you in 3D how the molecules that we've been looking at in cartoon form are actually working on the surface of antigen presenting and T cells. I'll show you both how antigens are presented and then the down-regulating interactions that occur with CTLA-4 and PD-1 and how those can be interdicted with monoclonal antibodies. And then I'll give you a very, very brief primer on how to access the protein data bank. We are always interested in traffic. Those of you who were in the hall before the symposium began will see that we had a control panel there that showed you how many people were using the protein data bank website at that precise moment, who had been from what city in around the world using and the United States using the PDB. And then I'll make a second plug for the boot camp and for the new Cryo Electron Microscopy facility that is in the final stages of commissioning. So the Protein Data Bank was the first open access digital data resource in all of biology. It was founded in 1971 by structural biologists as an open access resource before anybody even knew about the concept of open access. There were 7 X-ray structures of proteins available at the time. And these went into a communal data store first at Brookhaven National Lab on Long Island. And I think that the willingness of structural biologists who were X-ray crystallographers at the time, who were confronting the challenges of trying to understand how to move the field forward from its infancy, had a critically important effect on accelerating the growth of structural biology. I don't think that we would be where we are today in crystallography, NMR spectroscopy, or electron microscopy without the open access data sharing that began voluntarily in 1971 and became compulsory in the 2000s, both depositing the atomic coordinates and then subsequently primary data underpinning the structure determination. Shown here on the right are some of the very first protein structures to be determined, myoglobin being the first, and then followed fairly soon thereafter by hemoglobin. And then, of course, there were a series of enzyme structures published, the first of which was lysozyme from the lab where I did my D.Phil at Oxford. Today, the archive has got more than 123,000 entries. There were 100 when I started graduate school, under 100 when I started grad school in 1980. The archive, instead of being focused exclusively in the US, is now a global activity, and there are 4 regional data centers, one in the US, one in Europe, one in Japan, and there's also a specialist NMR data repository in Madison, Wisconsin, the Biomag Res Bank, or BMRB. So together, what we do is we manage the archive, and we collaborate with each other on bringing in all the depositions and archiving the data to a common standard. And then we compete on showing different views of the data. We compete at the level of the website. The US data center for the Protein Data Bank moved to Rutgers in the late 1990s under the leadership of Professor Helen Berman. And there are actually two sites at which we have PDB teams. One is here at Rutgers, just down the hall from where we're sitting today. And the other is at UC San Diego. The importance of that is all the Asia-facing internet traffic that we have to support, serving up our data to people around the world. 1.5 million atomic sets of atomic coordinates are downloaded from the archive every day of the year. It's over 500 million a year. There are north of 1 million unique users of the PDB in any given year, and approximately 200 data resources in biology actually repackage and redistribute PDB data. So this information is reaching everyone doing biomedical research, whether it's in academia or in industry. Worldwide, the industry people are maintaining copies of the archive inside their firewall and interrogating them and merging them with their own data stores of 3D structures determined inside. Our job is to serve up all this information, to safeguard it as the archive keeper here in the US, and to do outreach and education to a very diverse set of user communities. We know that about 3/4 of our users are not structural biologists. They're biologists, clinicians, students, educators, people involved in doing research. So much of our effort is aimed at democratizing access to what is quite complex and technically sophisticated data. You can see most of the people involved in the PDB today. They were here in May of 2013 when we reached the 100,000 structure mark. A lot of smiling faces there, both on TV screens and here at Rutgers. So the contents of the PDB are incredibly diverse, like our user population. It ranges from very, very small enzymes, hemoglobin, myoglobin, things like that, to very large viruses, the largest of which is the Fausto virus. There are integral membrane proteins, structures of which have been determined either in lipid bilayers or in detergent micelles, many of which are depicted here in this image. There are proteins involved in muscle contraction. There are proteins involved in constructing microtubules. The ribosome, the proteins that are involved in making the pre-mRNA from a gene, proteins involved in DNA replication. All of these are represented in this incredibly diverse collection of biological macromolecules in the PDB. Many of these are drug targets with small molecules bound. Others are drug targets with FABs bound, where there is actually a monoclonal antibody treatment. And that's what you'll be seeing today from me. And I think a little bit later in this afternoon's presentation, Dr. Jack Hunt from Bristol Myers Squibb will probably say something about small molecules. And I hope he'll show some structures, but we'll see. So continuing with the 3D theme, here's an artistically colored rendition of a T cell interacting with an antigen presenting cell. This is an electron micrograph that's been colored to highlight the two cells. And the role of the antigen presenting cell is to, with the MHC molecule that's sitting on the surface of the cell, right under here in this immunological synapse, is to present these short peptides, one that Dr. Latimer was talking about just a moment ago, to the T cell. And the job of the T cell is to answer the question, is this me or is this somebody else? And if it's me, there's a mechanism by which the system avoids mounting, in most cases, avoids mounting an immune response to self. But if it's a viral antigen or a neoantigen from a tumor, a mutant peptide from a tumor that the system hasn't seen before, then of course you're going to get an immune response. And this is depicted, the antigen processing that occurs in the antigen presenting cell is depicted schematically here. The subject being a virus, and the viral proteins are actually degraded by the proteasome into short segments. And then these short segments can make their way into the endoplasmic reticulum, where they're picked up by the MHC complex, which is actually two proteins that form a non-covalent partnership, the beta-2 microglobulin and the MHC. And these transit to the surface where they're exposed, where this combination of two proteins and the short antigen are exposed on the surface for interaction with the T cell receptor. So at higher resolution, you can see what's going on here, where the antigen presenting cell meets the T cell, and the antigen is presented to the T cell receptor. One of the triumphs of protein crystallography was to explain this process. Some of that work was pioneered by the late Don Wiley, who determined the first structure of an MHC with an antigen in the cleft here. So the bottom of the screen is the antigen presenting cell surface. The top of the screen is the T cell surface. The green molecule is the T cell receptor, and the sort of mustard brown is MHC, and the red here is the beta-2 microglobulin. And sitting in this cleft between two long alpha helices is the peptide that's being presented to the T cell receptor. And it's this detection by Braille that's going to determine whether or not there's going to be a reaction of the T cell to the presence of this particular antigen. And we understand this process at literally at atomic level, and we can understand why some MHCs present some peptides and not others, and the way in which the T cell receptors are able to read out the amino acid sequence of the peptide and determine self from non-self. It's an exquisitely sophisticated process. But of course, that, as we learned earlier today, that's not the sole determinant of whether or not a T cell is turned on or off. We have what's known as the two-signal hypothesis of T cell regulation. This is depicted here. So if you get a presentation of antigen in the context where there are proteins on the surface of the antigen presenting cell that can interact with CD28, you're going to get a stimulatory response, and the T cell is going to react accordingly. Alternatively, as we heard from Janice's talk, if these, in fact, very same molecules on the surface of the antigen presenting cell encounter CTLA4 on the surface of the T cell, you're going to have an inhibitory downregulating effect on the T cell. And remarkably, we can actually see this in 3D. So again, the bottom of the slide is the surface of the antigen presenting cell. The upper surface is the upper end of the slide corresponds to the surface of the T cell. And what you can see here is the B7-1, a dimer of the B7-1 protein interacting with two copies of CTLA4, which would be projecting down from the surface of the T cell. The act of binding transmits a signal through the membrane, through this plasma membrane of the T cell, and there's a series of signal transduction events which serve to turn down, to downregulate the action of the T cell. There's a lot of molecular circuitry north of this image, and I suggest that there are molecules there that represent bona fide drug targets for small molecule interdiction of the impact of signaling through CTLA4. I should have mentioned earlier that every structure in the protein data bank has a 4-character ID. They usually start with a number, and then there's a mix of numbers and letters thereafter. You can actually, if you know what this code is, you can immediately go to the protein data bank website, rcsb.org, and actually view the structure in detail, as I'll show you towards the end of the presentation. Now, we heard about the success of ipilimumab, quite a mouthful for someone who isn't an immunologist. And here in 3D, courtesy of Steve Almo and co-workers at the Einstein College of Medicine, is a still image of the FAB fragment of this monoclonal antibody, the anti-CTLA4 antibody, interacting with CTLA4. And what I think should be apparent to you is that the part of the protein that's interacting with the CDRs, the antigen recognition sites on the FAB, is actually slightly different from the part of the protein that interacts with B7-1 or B7-2, when the two cells meet. So the presence of an IgG with this type of an FAB at its business end is capable of interdicting this downregulating response to the presence of B7-1 and B7-2 on the surface of the antigen presenting cell or on the surface of the tumor cell. There's another, as we heard from Janice's talk, there's another set of interactions that also inhibit the activity of T cells, and this, of course, is the PD-1, the programmed death one with the programmed death 1 ligand, PD-1, PD-L1, PD-L2. And again, we know about these interactions in exquisite detail at atomic resolution. All of this is in the PDB. In this particular case, the four-digit code is 3 Bravo India Kilo, to use the NATO alphabet. It's this binding event that occurs when PD-L1 meets PD-1 at the interface between the two cells that has the impact of downregulating the T cell. So how do the FABs work that block the PD-1 PD-L1 interaction and block the signaling downstream of PD-1 in the T cell? You can see here a 3D rendition of a very recently published structure from Merck showing pembrolizumab or Keytruda binding to a particular loop on the surface of PD-1 and effectively blocking the interaction of PD-1 and PD-L1 and hence the downregulation of the T cell. So we understand these things in exquisite detail, and of course we have a great deal of knowledge of what's north of this image. What molecular circuitry is it that actually turns the T cell response down as a consequence of the interaction of the joint interaction of the T cell receptor with the MHC with the antigen and PD-1 with PD-L1. So how do you actually get to these things? If you go to rcsb.org, there's a search box that you can type things into. In this case, I've just typed in the 4 character code corresponding to the pembrolizumab PD-1 co-crystal structure, and you can go from there to what's called our structure summary page, which tells you everything you would want to know about this particular 3D structure, and in many cases a lot more. There's a great deal of information here of interest to structural biologists, but the bulk of our effort, as I indicated earlier, is going into creating functionality on this structure summary page that will be of direct interest to biologists and people studying how these molecules work in cells or work in the circulation. So before I close, I just wanted to give another little plug for the boot camp and for the single particle cryo-electron microscopy facility that's been established here in the basement. When the building was built, a cryo-EM suite was part of the design. There is space for 2 microscopes, a junior entry-level microscope. A junior entry-level microscope today costs $3 million. Or the high-end, top-of-the-line Cadillac, Mercedes-Benz, Ferrari, whatever you'd like to call it, that costs about $5 million. So thus far, we've been able to outfit the smaller of the 2 rooms for the $3 million microscope. It's been delivered, it's been installed, it's been commissioned by the manufacturer FEI, and we've hired a very experienced PhD trained director from the Center of Mass of Cryo-Electron Microscopy at Martins Ried in Max Planck Institute at Martins Ried in Germany, just outside Munich. His name is Dennis Thomas, trained at Brandeis and at MIT. And he's the one who will be finishing off the commissioning and then working with the users. Initially, most of the support that we're going to be providing is for the so-called single particle structure determination. So you're looking at individual particles on the EM grid, such as this virus, by taking many, many images of viruses and of this particle in different poses. It's possible to computationally reconcile those into a 3D mass density map, like an electron density map measured in crystallography, and then to interpret that in terms of an atomic level structure and to be able to overcome the barrier of being unable to crystallize most of these things. When we made the order for the microscope last year, we understood that there was probably a sort of a lower limit in terms of the size of the sample below which it was not going to be useful. And that lower limit was somewhere between 100 and 200 kilodaltons. But very recently, at a symposium in New York City last week, someone stood up and presented a near atomic resolution structure of the hemoglobin tetramer, which is only about 70,000 daltons. It relies on a brand new technology called the phase plate, which actually increases the contrast between the protein that's embedded in the layer of vitreous ice on the EM grid and the surrounding vitreous ice. And it's that contrast enhancement that seems to offer up the possibility of doing cryo-electron microscopy even on relatively small proteins, and hence putting protein crystallographers out of business, except those working in industry where the questions are very, very high resolution. So there'll be a formal opening, announcement, etc., and a formal initiation of operations of the cryo-EM facility. We will at a later time be offering up access to being able to do tomography, either on individual cells or even on tissues, again embedded in vitreous ice on the grid. The way in which we're going to try and set our users up for success with the facility is to provide an opportunity for this crash course that in the first week is going to allow people with samples to bring them forward and have them characterized both biophysically and in the cryo-EM to determine whether or not they're reasonable candidates for study immediately or whether it needs to be more work done on generating the sample. Then in the second week, and this will be opened up to a much larger group of registrants, we're going to be going all the way from those recorded EM images through all the software required to actually get to a mass density map and an atomic level interpretation of the 3D structure. So that'll be done in the second week from January 9th through 13th. The preliminary studies of the samples will be done from the 3rd, 4th, 5th, 6th. Yes, 3rd, 4th, 5th, 6th of January. And the second week's actually open to participants from industry, and there'll be flyers going out to all the structural biology groups in the pharmaceutical companies in the immediate vicinity advertising that opportunity. So let me finish by again acknowledging the enormous number of people who work at the Protein Data Bank who make access to those 123,000 plus structures possible. I also want to acknowledge the partners that we work closely with on managing the archive, the Protein Data Bank in Europe, Protein Data Bank Japan, and then of course Biomag ResBank in Madison, Wisconsin. We're funded jointly by the National Science Foundation, the National Institutes of Health, and the US Department of Energy. It sounds like an awful lot, when you actually see the size of the annual grant, it sounds like an awful lot of money, but in fact, it's not nearly enough to do all the things that we have to do, because not only do we have to manage the archive, manage all the incoming data, but we also have to get ready for the future. And the future is the combination of cryo-EM, the electron microscopy studies of large complexes with data coming from other sources, and to be able to build a data archiving system that includes not only data from EM and crystallography and NMR, but data from things like chemical cross-linking, fluorescence resonance energy transfer, homology modeling. All of these different tools are being brought to bear on what's called integrative or hybrid methods, which allow for slightly lower resolution, but nonetheless incredibly penetrating studies of very large macromolecular complexes. There's no way you can crystallize them. There's no way a single technique is going to give you all the answers. But we are on the threshold of some amazing advances in structural biology, not just because of cryo-EM and the phase plate and direct electron detection, but because of the way we're going to be able to combine data coming from different sources to study very, very large, very exciting macromolecular machines that underpin basic biology and, of course, human health and disease. Some of them are even drug discovery targets. So with that, I'll close and thank you. And I'll warn you that we are going to take a big group picture outside. The weather is actually cooperating today for a change. We want to mark the event. And I think we're going to do that straight away, right? We're going to go out and take the picture. And then there's going to be coffee, refreshments, etc. served. And then we'll resume at 3:45.